ended up studying stem cells in plants, did that for a number of decades in Arabidopsis, very fundamental. 7 years ago, decided that it would be very interesting to see how fundamental knowledge is actually used in practice. So I moved to a Dutch breeding company, family owned, big budgets, no shareholders, lots of opportunities. And I wanted to share a few lessons that I've learned from having looked at both sides about all the ideas that we have about helping breeding by having excellent plant science. And the title has changed, but the opportunities and pitfalls have remained. So what would I like to go through with you, and you can see from the background colors of the slides as we progress through the talk, is first of all, I would like to just present you a story which is just like the story after it, from the perspective of breeding is a failure story. But as we know from Karl Popper, you learn most by disproving hypotheses. And so I think there are some very important messages from these, let's say, failures from the breeding perspective that I think help a lot to see and to what is the path to the future in order to appropriately use, let's say, knowledge that we assemble on genetic networks and on the functions of genes and proteins in our models and in our crops in order to design the phenotype from knowledge of the genotype, which is what we really like to do. Knowledge of the genotype is easy these days, but connecting it to phenotype is the main issue. So what did breeders do? Basically, they adapted a selected DNA variants from a number of plants which looked promising in the past, and here you see to the left the ancestors, and they made them into an incredible variety of things that had never existed before and which we now eat. And there you see the assortment of the company that I work for. There are very many different ones, and they are vastly different from the wild ancestors. And in fact, what has been done therefore is a beautiful demonstration of what Darwin first found. Actually, by looking at domestication, he got the idea that selection allows you to modulate the genetic content such that you can actually try to exploit what you see in variation and select for it and come up with the desired phenotypes. So that's what breeders have been doing, and I should mention here it's terribly successful. And so we as plant scientists should see where we have the right role to actually further improve that process. It's not like we have to invent something new. We have to help to improve this process, and for that we can use our ingenuity. And of course, there is a number of very recent breakthroughs, if you look at my timeline, which actually build a real promise that we can much more have an engineering view on how to change DNA so as to improve our plants. We all know the advent of genome editing, and it is under everybody's attention. And there's a huge promise that from being able to so specifically manipulate DNA that you can actually, at the very least, speed up improvements of varieties in breeding, but maybe also think of new things which would not be possible with conventional breeding methods. And the second thing is, of course, even more recent. We get tons of protein structure information by inference now by using the wonderful tools like AlphaFold and all the clones and varieties that have come on the market for that. And that also gives a great perspective, because right now it's a bit too early, but you see the time coming when you can use these tools to also do a functional prediction on what a gene is actually doing in terms of the protein that it encodes and how you need to change it in order to make that function slightly different or very different, which now we have to rely on variation in nature to actually learn from what these capacities are. But there is this promise in the air that at someday we may be able to calculate how to change the protein in order to change a trait. So those are all big promises. And let's look at this prediction thing. Because there are two things now. There's a new wave in plant biology which says soon we will be able to predict the phenotype from the genotype, and we can then help. And I would like to look at that predictive thing. And here's the reason why I joined the company, because I feel very sympathetic to a quote that is attributed to Richard Feynman, which may be slightly misquoted. It says, "What I cannot create, I do not understand." And this is in the specific context of physics, but I think it's also true for breeding. In breeding, we have created a lot of things, but we rarely know why they are the way they are. And I think the gap there is that we need the fundamental knowledge to have models, to have to predict what is going on. And so this is really analytical and predictive science. But I also want to mention Theodosius Dobzhansky in this quote that was actually used in a slightly different form before, I would say in a reduced form, but that we can discuss later, where he says, "Nothing in biology makes sense except in the light of evolution." And the background of that statement is that biological mechanisms were not put together by an engineer who tried to isolate the different functions in different parts. They were tinkered together very often by functional sharing, so that biological systems are completely intertwined and cross-wired, not with the purpose to separate the function in separate blocks so that you can replace one part and put in another and change just one function, but you have the total you have the total system to work with. And if you change something drastically in one part, as we have seen several times during the past days, you get changes in other processes. And we have to deal with it. So the question now is, how can we bridge these two things? So far, breeders have used selection. And that's using to the maximum what evolution provided. Can we also bring in analytics? Because if we say that gene editing is going to revolutionize breeding, we must actually also know which genes to edit, and we must be able to accurately predict what the effects are. My hopes are high for disease resistance, and I hope that Pam will prove me right after the break. But I think it is difficult in many, many areas. And that's why I come with these two stories. So if you talk about prediction here, this is, some of you know these very well. They may even have some warm glow inside. You look at the Maxwell equations, you see 4 separate equations, of which 3 of them are based on experiments and the other one was based on reasoning. But you see here something that we often forget in biology. You have separate relationships, but the relationships depend on each other and they all mutually crosstalk. And you need very proper analysis and beautiful mathematics to actually come from these, which are just 4 equations, to all these applications that we have in the modern world, which depend on electricity and and the early forms of electronics. So that's predictive power. The question is, can we develop a similar predictive power in biology? Because if we are to design plants to make them resilient to whatever stress, for example, we have to understand how the different subsystems connect to each other, just as these different equations connect to each other. Now, how comes the case study? So this is actually the work that ended me up in the company, because we started this when I was still in university, and we started a collaboration on the positioning of leaves, phyllotaxis. And we found there that 3 transcription factors, which are also involved in stem cell programming, that's where we come to it, if you mutate them all 3, actually give rise to a different phyllotaxis. And we quantified that phyllotaxis. You see that to the right. So you move from a spiral to very frequently 180 degrees juxtapositions, but it's not completely stable. You see that in these diagrams, which is previous angle versus next angle. You see that in the normal phyllotaxis, the golden angle is the red dot is actually the highest propensity, and you see a distribution around it. And you see what our mutants do, they shift this, they make it more variable, but it finds, as it were, a new attractor, which is the 180, 180, which is in the right bottom corner. It's not as high yet because it's metastable as in the wild type, but it gives you the idea. We published this idea, of course, that you can shift phyllotaxis, and you can, for example, make a cucumber which has all the leaves in one plane and all the fruits elsewhere, so that harvesting would become so much easier, which is extremely relevant because we have no people in Europe to harvest our crops. Lo and behold, if you mutate the paralogous genes in cucumber, and this was done in the company at the time, by using tilling, you actually get similar shifts and you move to the metastable changes in the 180-degree pattern. So that looks quite promising, right? And it's usually the kind of information that in breeding companies, people use to really go full force and say, well, this has been shown to work in academia and we can transfer it to the crop. It has been shown, we can go for it. But let me tell you first how we try to deal with this metastability. That's by using actually random variation, not by nature, but by creating random mutations in the genome of this triple background with the idea that this plant is metastable because we have just not pushed it far enough in the right direction to spiral down in the attractor of the 180 degrees. So we need somewhere else, we don't know where, we need another mutation and then we will stabilize it. It's quite a reasonable idea, right? You can already feel this coming. But I would say it's a reasonable idea. Now you do that and you do a very big screen and you find no suppressors of the metastability which create 180-degree paths, but you only find enhancers in multiple genes. Beautiful enhancers, because what you get is kind of a snail's hole in the meristem. That's the only thing that this plant makes. And what you see is that it actually spirals out. It has no notion anymore where to position its leaf. So it's a completely, it can still grow, but it is completely confused as to where to put the leaves. What is the gene affected here? A number of these suppressors we cloned out by methods that are not important here. So what we found is that there was an allele of a polar auxin transporter which fitted completely with our previous ideas of how this gene worked, but it was a very, very subtle allele on itself that totally no phenotype. So this shows that this pathway indeed makes very strong use of polar auxin transport, because even an incredibly small reduction in capacity of a protein by a single point mutation can completely render the system unstable. However, it also showed us that we have been thinking wrongly about metastability. Everything fitted because these three genes actually bind to the promoter of PIN1 and thereby slightly reduce its expression levels. It's direct binding, so everything is direct. So scientifically, it's perfect. But breeding-wise, it's useless. So we didn't find any stabilizer. Moreover, when we moved from Utrecht, which was the place of success, to Wageningen, the place of failure. Because what we saw is that even in our triple mutant, which had this nice metastability, the metastability was lost under new growth conditions. No way to recover. Everything was tried. We went back to Utrecht, but the climate room was changed, and we could never get back. So what you see is that there is an incredible, let's say, environmental sensitivity to this phenotype. And we were just lucky the first time that we found enough stability to see the phenotype and to see the function, but the function is you cannot sell seeds on the premise that you will have your leaves in the right position. Now, what is going on, that was very much helped by inspiration from very beautiful work in Olivier Hammon's lab, who is not yet here, I fear, but may come later, who actually saw that some things that they could not explain about a genetic interaction could be explained by the fact that there is a torsion of the stem, and the torsion of the stem actually displaces the leaf primordia relative to each other in combination with growth in length. And lo and behold, when we looked at our mutants, and I won't go over the precise data, we saw that same torsion. You actually see that you get different effects in the phylotactic pattern, because the torsion is always leftward, but the phylotactic spirals can be either to the right or to the left. So it means that you get two different patterns. In one case, you can make 180-degree angles, and in one you get more 90-degree angles, but it's noisy. But if we then looked exactly what happened in this triple mutant, you see that the internode length changed, the plastocron, so the timing was exactly the same. The torsion is also the same, we measured that. And what you see is if you put this all in a very simple mathematical model, which just puts the initial position of the primordia and then does the right growth characteristic, you can actually recapitulate. And you can also see that you only get the 180s under a certain, in the counterclockwise spirals, which is an uncontrollable system because that is a purely stochastic system, which depends on very sensitive initial positions. You could never do something like that. And indeed, if we then made real torsion mutants, we could very easily show that you can move the torsion mutants in one particular direction of the clock, which is one edge of the diagonal. We can move them to very frequent 180, 180. But now we have another problem because these mutations which change torsion, they also change everything else in the plant. So you move from one pleiotropy to the other. And very clear conclusion, phenotype predictions based on the first paper that you see when everybody jumps on it, are oftentimes wrong. And only by understanding the complete system, by doing a lot more work, you can reach the solid conclusion about what kind of inputs are needed to make a certain result. And there are more than you initially thought. First you think, well, it's a couple of transcription factors, piece of cake. But doing all of this together is basically impossible because you will never get rid of the clockwise and the counterclockwise turning frequencies of the spiral. So it's a dead end. It will nevertheless probably be a nice publication. But you see that the premise that if you have a certain body of knowledge on what genes do, and then directly want to jump into the conclusion, we now change this in the crop, if we find the autologous genes and they do more or less the same, and then you want to apply it, there are many traps in which you can fall. All right, that's story one. Now story two. It's about trait complexity. I would like to bring you into the world of pepper pungency, a very visible thing. There are very many peppers of very many different shapes and forms and so on, and they're all very different in pungency, except of course our sweet pepper, who has a pungency of 0. What causes pungency? This is the most simple case of breeding genetics. It's just one enzyme, capsaicin, at the end of a complicated metabolic pathway. It is really the core step. You have no pungency if that enzyme is not active. And that was found a long time ago because and it's an ideal case for the breeder. If you want a non-pungent pepper, ultimately you need mutation in only one gene. It's a very specific gene that only has this one function. And if you knock it out, as was done by, let's say, mapping this gene and finding that there is indeed a disruption, let's say, in the promoter region of that gene, which classifies it as a null mutant, and then you get the things that we have in our bell peppers, which is 0 pungency. So that's completely easy. But actually what you want is not always zero pungency. You would like to glide the scale of pungency. Some peppers are so pungent that majority of people cannot eat them. And yet they have wonderful tastes, which are actually hidden behind this, let's say, assault at your heat perception system. And therefore, you would like to turn the dial. And now we're going to see what that requires. First thing is to see, okay, we know exactly which enzyme it is. We have AlphaFold, we have structures. We just modify the enzyme to tune the level of pungency. That's actually very, very difficult, because you see that in these in these different pepper varieties, which have very different pungencies, there is very strong conservation in the reaction channel and the actual reaction site. And you have not really good clues to engineer for a very specific amount of pungency. Because many cases, when you tinker with these amino acids, you do not only make the enzyme give it a different activity, but you also change the access of the substrates, and you do several things at the same time at the molecular level. So that's hard. Then you could say you do it by the RNA. That's very difficult, because in the end, and here you see all of the enzymes that are linearly connected, let's say, to this final step. And you see in a paper that was published more than 10 years ago, you see what are the expression levels of these different biosynthetic genes. And you see that there is a wide variation, and they are coordinately required. And actually changes in any of these activities will feed through to the end product. So you do not have to juggle with the levels of just one enzyme, because in all these different pepper varieties, levels of these enzymes are different. So if you make crosses between cultivars to make something, you mix up the information there. And so you have to deal with this fact that there are many enzymes. And the other thing that you have to deal with is that capsaicin is not made everywhere. You see here a capsaicin antibody. It's a very old picture, but I love it because it makes a very clear point. You only have the capsaicin in one layer of cells, which is there in the red block in the interlocular septum. And peppers have huge variation in the number of tissue that they have in the shaped fruit, which actually contributes, of course, then to the levels of capsaicin. And that means that in order to control this level and control it, you have to control also the contribution of the genetics that is in fruit formation. And here I show some diagram from Arabidopsis, which has many handfuls of genes, which all affect the shape of the fruit. And you can imagine that in pepper, all the counterparts are also in some form there. And there is allelic variation between these. And so if you make a cross between lines in order to preserve, let's say, a certain level, you will have to know what is the allelic status of all of these genes, because all of them influence pungency. And in general, you can actually see, if you look at the shapes of very pungent peppers, there are actually this correlation to the importance of shape is actually there. Finally, you have fruit ripening. And there is an endogenous pathway for fruit ripening, and variations in all of the genes involved in that pathway, in the developmental pathway, will, of course, affect the ripening, and therefore the amount of capsaicin, which is only made at the later stage when the genes for the enzymes are activated. But it also has a very important link to, for example, ethylene signaling, which is notoriously noisy and completely environmental dependent. And what my pepper breeders say, if they do the capsaicin measurement, sometimes they have a branch of a pepper which hangs to the other side, and there's a total different pungency in the few fruits which are there than from the fruits that are hanging there. And if there is a stress factor, or if a disease comes by, everything affects it. Now we make up our list. We have the black ones that we discussed, But of course, you know, if transcription factors are involved, in every case where you look for modifiers of transcription factors in any developmental biology system, you find all the RNA stability factors, DNA methylation factors, chromatin modification factors. So allelic variation in each of these genes is in the end also going to affect pungency. And the allelic variation is there, because if you cross different varieties, you mingle together in an immense amount of allelic variation. So in the end, this very simple trait becomes a complex trait, which you might be able to shift, but not by changing one allele or gene editing only the capsaicin synthase. You have to coordinately follow all the alleles that have effect, which have a wide variety of biological process, and then move it to some other optimum. You will always get variation because it's a normal distribution. There are too many genes to get single effect factors or QTLs. You just juggle everything together and you do genomic prediction, which is that you do statistics. You do a smart way to rapidly try to move this peak somewhere else. But you do never arise at our ideal having a variety which has just this pungency and which is only at the mean and slightly above without having these white dots. Now, I've given you quite some food for thought about what can go wrong if you want to implement knowledge in breeding. Now I also have to probably offer some kind of a solution before. And I think we can definitely do something, and I'm trying to make a synthesis. And I have to draw in what we know about multigenic traits. I have to bring you back that phyllotaxis, the first example, we only talked about 3 genes and a few others that affected them. But of course, the same reasoning holds there. In the end, that's also a multigenic trait. So for very many things that we think are simply controlled, and not simply controlled if your genetic background is diverse. So then we go to the humans. And for decades, it had been very puzzling that if you look at height distributions and let's say you look at genetic effects, you in fact only find small effect genes, but you find extremely many. By extrapolation, there must be more than 100,000 SNPs in the human genome which has been associated with heights. That's many SNPs per gene. And it's what is called in their jargon an omnigenic trait. Almost everything affects height. But still they, over the last years, made some sense out of that, and that's useful for us breeders. The thing is that you have a circuit which is very important for growth, which is a circuit that operates between different organs, and there is a feedback between that, and the 2 factors are very important. That is the growth factor 1, and that is a pathway which actually has huge effects on growth. As you see, when you have mutations in these genes. So here are this is a very absurd, let's say, picture where people were brought together with some kind of fun about it, just by illustrating the enormous effects that you can get from what we now consider to be almost single allele mutations in these core genes of the height pathway. Obviously, these are not happy people as depicted here, but they're actually sick. They have a lot of syndromes associated with this condition. That's because this pathway is completely pleiotropic and used in very many circumstances. So if you interfere with this pathway, it's not going to help you. And it's very similar to, let's say, if you would very strongly influence a polygenic trait by one gene without making countermeasures in plants, my prediction would be you create a lot of pleiotropic effects. And there are quite a few examples of that, where that happens. It's funny to see that the same genes are also found in dogs, and there you can more easily show that these genes really have a lot of influence on the genetics, and they explain a huge proportion of the enormous size. But also here, of course, we have certain dog breeds which are, in fact, intolerable. And so there's also the same by effects there. Now, the kind of insight that came from very carefully looking at what kind of genes contribute to growth came from studies on QTLs combined with studies on causal genes, which were very nicely selected by looking at the genes which make stem cells, chondrocytes, work at your bone joints. And this is a recent study where you see that if you identify the genes which are active there, and then look at using a methodology that is called eQTL, look what the effects, whether you in populations can find, let's say, variants that correlate with these genes, you can actually come to this thing that was predicted already by Jonathan Pritchard a number of years ago. You have a set of core genes, and then you have satellite genes which are connected to them. In the case of bones, it's amazing because you have different satellites, and there is one satellite that operates in the lower part of this bone and the upper part of this bone to make them coordinately activated to grow. There's other coordination sets here, and there's coordination sets in the vertebra. Those are all different genes. They overlap a little bit, they hang around this core, and then there's many other genes which change, which are thought to have very small effects. And so you have a few genes which have a lot of effect, the pleiotropic core factors, and you have a surrounding circle of further and further away located, and here you show it as protein interactions, but you can also show it in a gene regulatory network because the same reason holds there. There, you actually see that you can understand that every gene is involved, because normally there is counter selection against genes which are very close to the core because they have too large effects and they have side effects which you cannot tolerate. But you can also reach a similar effect by jiggling a lot with all of the small effect genes and making all these effects going in the same direction. That's why we do what we do with genomic prediction. Why are such large scale effects caused by rare alleles? That's the simple question is because the proteins are extremely important. They do a lot of things. And you have found this gene and its protein because it controls a desired trait, but it does much else. So if you make a mutation in the gene and you make just a regular mutation, obviously you do not only block, change your or enhance your desired trait, but you also create a lot of side effects. And so in the case of these rare alleles, so here you create essentially a situation that is in many cases lethal or at least very severe. And you can, of course, mitigate that in one way, which is if you have such a mutation, you can have a very precise mutation. That's what we hope. It's actually what you try to do with the capsaicin enzyme, and it doesn't work with all proteins, but you might. But then you also know that these mutations are extremely, extremely, extremely rare because there's huge constraints on what type of mutations can select one specific output of that protein while leaving the others untouched. But there's a second thing that you could do. You could actually still have, let's say, you could still, let's say, get a specific block in the or change in the desired trait if the genes in the shell are also allowed to co-evolve, so that they actually compensate the change in the original protein and mitigate the side effects. And these effects have actually been found in several evolving systems where you can see that. But then you see, you are not depending on just a change in one protein, you are depending on changes in other proteins. So you either understand which proteins those are, which is what we do by all the beautiful work that Nick has been showing, but then you have to also know what to do with these proteins and how important the links are. And so then you could get to the control of your desired trait. Now, what does this mean for the future? I would say for very many traits, genomic prediction, which is the use of DNA markers for selection instead of phenotyping everything, which is very, very efficient in, let's say, quickly improving a population, looking at all the genes without knowing what the genes do. There is now a lesson that we have learned. There are core genes for every trait. There are peripheral genes. And if you understand how the core and the periphery connect, you can actually do something because you can start fine-tuning. First of all, large effect genes, large effect alleles are probably not there in your population because they have been counter-selected already in earlier phases before you have your progenitors for the cross, but you can make them. And then you can compensate their effect by making mutations in the shell, which is 1 or 2 levels deeper. And you may be able to make something which very rapidly moves your trait towards to a different, let's say, value in a desired direction and compensating for the by-effects. So I would say that very many cases, my prediction is that very many cases in the future where we use gene editing to effectively make trait changes without affecting other side effects require this knowledge that you as academics, and actually also me 1/5 of the time, are assembling and building into this body of knowledge in which we know how to use. But we cannot do without this part of the work because there is just so much unknown. And I try to make the point clear that there is no such thing as a simple trait in the, let's say, in the context of the variation that you have in a breeding pool or in nature. Every trait is polygenic. And in that sense, I think we need to bring these two fields together. So what are my conclusions? 1, we need in-depth biological knowledge to predict the effects of gene edits. So we don't need to just focus on being good at gene editing, but we need to focus on being good in biology. Second, the gene edits in many traits must be balanced to allow predictive breeding, to allow to be able to use large effect, but to compensate their side effects. I think we are assembling information that may in the future allow us to do so, but there are very few cases in which it can actually be done. And maybe the gene for gene relationships in biotic stress are the first place to try to do it. And the third, which I already wrote here before yesterday afternoon, I think bringing it back to the Anthropocene, all of this is jibberwocky if we do not develop a joint sense of ecological awareness, solve geopolitical issues, and in that way mitigate the major effects of climate change. Because I do not think that just by doing the science, we will get anywhere near. Thanks for your attention.